Swayam Prabha. Digital India. Educated India.
Yeah, good morning. I welcome you all to this lecture in this course on analytical, spectral and microscopy applications of inorganic compounds and nanomaterials. In the past two classes, we have been looking at the applications of mass spectrometry in this particular area. And now let us continue with the same mass spectrometry with another technique. So I have given you in the previous two classes the general glimpses, general ideas, general approaches of all this, followed by the more detailed aspects of electrospray ionization, which is simply referred as ESI mass spectrometry or ES mass spectrometry. So we looked at several examples of how to understand the spectra and these things. And now let us look at another technique which is equally important, equally useful and equally less, the fragmenting the species. So it's called soft ionization. Both the ESI and the matrix assisted laser desorption ionization, which is popularly known as MALDI. So let us look at this technique of MALDI in this particular class along with the principle as well as the applications of it. So in this case, as you can see, the matrix assisted. So that means there is some kind of a matrix. The matrix is supporting, helping, doing something, and it is a laser. When you apply the laser radiation, something happens to the matrix and the matrix helps the molecule, and that leads in the desorption, and the desorbed molecule will get the ionized, and the ionized molecule is being analyzed. So I hope you understood the meaning of matrix assisted laser desorption ionization, how it is pertinent to that. So having understood the meaning of each of these words, then if you combine them, so what it gives is basically you take your compound in a matrix and you put some laser radiation, some suitable laser radiation. What is suitable? We'll look into details in a while, a little later. And this suitable radiation, when you incident on the sample, the matrix will dislodge the molecules as well as the matrix also. So variety of species comes into the comes out of, that is what is basically called the desorption, and these desorbed species are further ionized where you get the ionization. Now kindly please look at the slide, explains clearly. And such kind of technique is useful for bigger molecules, large molecules like polymers, polypeptides, proteins, nucleic acids, carbohydrates, variety of biological systems, synthetic systems, somewhat complex small molecular systems. All of these can be studied. The important thing is that you can ionize these without disturbing their intermolecular interactions. That means if the proteins are associated as a dimer, trimer, tetramer, you can still find, even compared to ESI method. So it is more or less equivalent to ESI method, maybe even better in terms of the ionization process, that is much more softer for the MALDI as compared to the ESI, which makes it a researcher to look at the details of the molecular aggregation, even in proteins, peptides, etc., or any biomolecules, without their dissociation, and that's a very good factor compared to that. So please have a look at the sample which is put into the matrix, and then you put the laser simply, and then the sample along with the matrix units will come, and then you separate them, you ionize them, and then you take into the detector kind of thing. This is one way, very simplistic way. I will show you a little bit more elaborate way in the next slide for a while, after telling you some of the steps that we are looking at in this. See, assume these hexagon kind of a blue shaded ones as the matrix, and these ellipses which are shown as the species, could be positive, could be negative, etc., and then when you irradiate with this light, light is shown as the yellow. So you can get a species of the negatively charged, species of positively charged, species of the analytical molecule which is associated with the different extents of matrix, all of these kind of things. Then what you do, then you go into the ionization chamber. Then once you ionize, you take into the detection chamber, and that's what it is. And these are the things which are written as step 1, step 2, step 3, step 4, etc. So you have a simply ablation, the ablation desorption to ionization. So what is this dependent on? On your the desorbed species, ionized species, these are all dependent upon the nature of the matrix that you're choosing and the intensity of the laser that you're choosing. Okay, the matrix absorbs the laser, not your molecule, and that energy is transferred to desorb the whole system. That is where the thing is there. So, and the desorbed one will have a neutral species, ionic species, some non-dissociated aggregated species, their ions, and the matrix species, clusters, all these kinds of things. And that's where we look at. Yeah. So whatever I mentioned to you earlier, it can be seen with more diagrammatically. I put 3 different diagrams. All I want to convey is the same from all 3 of them. I'm sure one will be able to understand either from one of these or the other. Look at the top left panel in this particular slide. You can see this is the template, which means which has got the compound and the matrix. Here the matrix is shown by the greenish species balls, and the red one is your analyte. Now what you're applying a laser beam on that, and the laser beam does the ablation and desorption. So the desorbed will have a variety of species, shown only one for example, but you will have variety of things. From here you can have a desolvation ionization, or add a proton and go for ionization. So therefore you will get differently charged species also. And then once you ionize, you take to the mass spectrometry. That's the conceptually. So the same thing is shown here, and you have your the sample along with the matrix and the beam light, and then you have differently charged species, and then these are further ionized and then allowed it to go into the mass spectrometer. So, and these things can come out as a time of flight. So you have your sample, you have your desorbed, you have your ionization, and then you have an electric field which will defan these things, and then you have a time taken to reach the detector. This is the detector, and this time, this length will be traveled by those species what you are measuring, because you applied the electric field on this. So you have different levels of the time that they spend in this chamber. So that is what is called time of flight. So there is nothing other than that. So time taken for each of the species is different. So each species comes differently on the detector, and detector will give you the current. Of the whole principle of the MALDI instrumentation, I hope you understood. More details of electronic details of instrument is not required for this course, not only in this, even in other techniques as well. So now let us look at some examples. Before we go to the example, let us look at some matrix we mentioned, the matrices that are used. You see a molecule of this kind, the 4-alpha-cyano 4-hydroxycinnamic acid, cyanopic acid here, cinnamic acid, cyanopic acid, and cholinic acid, acrylic acid. So many of these you can see, all of them. All of them are acids, carboxylic acid. All of them have got the pi systems, you can see aromatic systems, and many of them have got even the aromatic, the heterocyclic aromatic things, etc. So that should reflect in our mind. So that means there is a carboxylic group, is again proton donor. The heteroatoms can also be as a proton donor, and the pi system can be a good absorber of the light, good absorber of the corresponding light, etc. And so therefore the matrix should absorb the light that you are incidenting using the laser, and then followed by desorption process, followed by adding the proton to that, etc. So therefore if you look at all of these examples are shown, there may be a few more, and each one of these will have a different excitation wavelength, and each one of these will have a different strength of protonation, etc. So therefore they are used for different applications. You can see using the lasers, and these are used for peptides, nucleotides, etc. This is used for peptides, proteins, lipids, then proteins. So you can sit and relaxedly go through all of these details. So, but I'm sure you understood what a matrix means, what its role is, and how it assists. So, matrix assisted, what does mean by assistance? This assist to absorb the light and then to dissolve the molecules. It assists to protonate the species, all of these kind of things. That is what is called the matrix assisted. Okay, now let us look at examples of this. You can use for protein in a mixture. Let's say when you do to take some kind of a cells, biological cells, and then you break those cells and get the material out and then separate the mixture, separate the fractions, then you will get a protein fraction. The protein fraction will be several different proteins. That means you have a mixture of proteins. Put to the MALDI technique, and you can see one example is shown here on the left top panel, and you see that this is one protein, this is another protein, this is another protein, this is another protein. So, it is a mixture of proteins you are getting because their mass are different. So, this is the myoglobin, this is the cytochrome C, this is the ubiquitin, and this is another kind of a cytochrome, etc. You can very nicely, and the area of this can be correlated with respect to the relative concentration of these in the protein mixture, in this protein mixture. So, that means now you understood application number 1. You can very happily, a mixture of proteins, you can identify those proteins, and this is a very good technique, and that is why it is used in proteomics. So, proteomic area in developing the huge amount of materials in the protein engineering, protein technology in this vaccine area. So, all of these where you use proteomics. Now, let us look at another example where the proteins form monomer, dimer, trimer, tetramer, etc., and see an example where such dimers or tetramers are not even broken also. So, they are intact. What it means is, so that means it is much better than even the ESI, and it is much better to handle high molecular weights beyond several thousands of the molecular weight. So, this is an example of the transferrin. Transferrin is a protein containing iron. So, that is where the transfers the iron ions across. That is not a part of this course, but we can understand that the blood harbor seals are the species. So, they are tetramer, they are dimer, and a monomer. You can see a monomer which is around this here, and then you have a dimer, and then you have a tetramer. All these kinds of things can be identified without breaking the aggregation, which means even if you have a molecular aggregation and forming some supramolecular aggregate, you can detect using MALDI even without breaking their interactions. The interactions are very weak. This technique will not break. That is why it is called soft ionization, the meaning of that. Don't take the literal meaning of the English meaning of the soft, but the soft here means does not even break their weak interactions. Look at another application in the same slide in the right half of the slide and showing a metallation. This is a protein of lactalbumin, apo-alpha lactalbumin. You started adding the mercury to little extent more, etc., etc., and then looking at the spectrum of the MALDI. Now, this is where the mercury is not added, only the protein is there unbound. So, you can see the full peak at one place which is corresponding to that protein, of course, at that purity. There is not. Now, you add 2 equivalents of mercury 2 plus to that, you would see something labeled as 1 and 2. That means this much amount of proteins have got 1 mercury ion, this much of protein has got 2 mercury ions, and this much of protein no mercury ion. No mercury ion 1, 2. Now, you increase to 5 equivalents, then you will find the without any protein, then 1, 2, 3, 4, etc. So, you can see this further and further, 10 equivalents. After 1 day if you wait, 100 equivalents, etc. You can see all of them making 3, 4, all these numbers shown on that is number of mercury ions to one protein molecule. Here you do not see unbound protein at all, completely vanished. So, entire protein is metallated, but to different extents. Some of them 3, some of them 4, some of them 5, 6, etc., and the intensities are there. So, that is the proportion you can make out. Now you understand, you take a protein and metal it, you can find out. Reason is one metal ion, that much of a mass increase, two metal ions, three metal ions, etc. All of these. I hope you understood all the applications that are shown in this particular slide. Not only that, in proteins many times you derivatize a specific amino acid or a specific type of amino acids. Let's say tyrosine, let's say aspartic, all aspartics, or some aspartic, not all, some tyrosine, not all. So such kind of derivatizations are known in biochemical processes where the chemist does. So here is an example which is shown for a cytochrome C, which is reacted with this particular molecule. Okay, this is in a short form shown as DBNBS. This is the nitro, then dibromo, and then sulfonic acid. So, and in presence of the hydrogen peroxide, and this reaction is done at different levels of ascorbic acid. Ascorbic acid 500 micromolar, 1 millimolar, 5 millimolars. So, different levels of ascorbic acid used. If you use A is here, A is for the 500, and then you have 1 attached by this one, 2 attached, 3, 4, etc. And if you have used 1 millimolar, then you have only 1 and 2. And if you use the 5 millimolar ascorbic acid, 1 and 2, but much lower in concentration, lower in area. So you have to see, compare with the protein area with respect to this area. Anywhere else, also the protein area with these ones, you can see more and more. So based on that particular conditions. So using the reaction conditions, you can always find out the extent to which the protein is derivatized. Here you are doing derivatization using the sulfonate moiety. Okay, these are simple proteins. So the cytochrome C, beautifully you can find out from this pattern. This is the cytochrome C and the mass. This is for ubiquitin, etc. I haven't shown examples here for ubiquitin. I have shown for cytochrome C. Okay, for the derivatization of this sulfonate moiety. Okay, so that is how one finds how much it is happened. Now let us look at another category of proteins called metallohistines. I am sure everyone has an idea about the metallothianins. That is, every third amino acid is a cysteine. Here metallohistidines is frequently you find the histidine as the residue. Now we know very much histidine is a very balanced amino acid. It can bind to a variety of the transition metal ions. This can bind to zinc, nickel, cobalt, copper, cadmium, mercury, etc. So these kind of proteins can be identified in the natural systems, plant metal binding proteins. Now here as an example is taken and shown for a zinc binding and a mercury binding. So this is the MALDI TOF mass spectra of analysis of protein metal complexes. It is a protein AGNT84-6, and that is what one of those kind of a metallohistines. And this particular thing is treated with the zinc 2 plus with using the resin. This is being taken through, and then at 1 millimolar of imidazole, these are separated, and then check the mass spectra. So increases in mass by multiples of 65. So because each 65 will add with 1 zinc, so it's the zinc mass. So binding from 2 to 6 atoms for zinc. So and then you can also see in the range of M plus 2 mass, etc., etc. So you can see that the 2 zincs, the 3 zincs, the M refers to the protein itself, the plus 2 refers to the 2 zincs, 3 refers to the 3 zincs, 4, 5, 6, etc. And then area you can take, and you can talk about the ratio of all of these. Absolutely no problem. Just like the example I showed you in the previous slide with the alpha lactalbumin with the mercury. Similarly here it is not that kind of a protein, it is a metallohistine protein where huge amount of histidine is being present in this. So similar titration which is done with the mercury, adding each mercury ion will add 200 Dalton. So therefore you can see that the mercury ion at that conditions of this conditions are given here, reaction conditions. What it means is, and you have very little of protein. M means not metal, it's the original protein without any metal. The M plus 1 means 1 mercury, M plus 2 means 2 mercuries attached, 3 mercuries attached, 4 mercuries attached. Okay, and that's how you can analyze and find out. So this is one way you can identify the whether they are the metallohistines and there is a large number of histidines are present, etc. By using any of these metal, you can use cobalt, you can use copper, you can use zinc, you can use mercury. An example is shown for the zinc and mercury. So it can also be detected, such metallohistines, when you isolate the protein fraction and then maybe further subfraction, you have the suspicion that this particular band is coming from metallohistines. That can be checked as well. Okay, in general, the mass spectrometry can be used in tandem, of course, means one mass spectrometry coupled with another, etc. Without coupling also you can get good information in the proteomics. I have been talking to you about the proteomics. Whatever I have shown here in this slide is a generic scheme. Kindly please look at the slide, and let's say you have taken some cells, you have lysed, you have got chromatographed and got some mixture of proteins. Okay, and that mixture of proteins, now proteins can be digested using the protein digestion, means the digesting enzymes. So that is proteolytic enzymes which will break the peptide bonds. Of course, there are several enzymes will break different regions of the protein depending upon what kind of amino acid is attached to that. The peptide bond where the cleavage is taking place, it looks for what kind nature of the amino acid is present. Okay, then you separate these ones, the different peaks, etc. Then you can put into the ionization chamber. Then you will get your mass spectrum for this, mass spectrum for that, etc. All the mixture, whatever you have, each one. Then you can pick up any one of these, this one, etc. Do further mass spectrum as such, so that you can do the mapping. You can do the sequencing also. You can initially identify the proteins, then you look for some specific sequence and then do further mass, that is called tandem mass. So, and you can use a little greater the ionization so that you can do that and then look at this. And then go into the literature, lot of data is available online and you can try to map them. So, mapping, I will explain that a little bit more better in the next couple of slides in this too. So, I hope you understand this. So, you can look at them, what kind of a proteins present in your mixture, and then in a given one of the protein, what kind of a sequence or what that protein is. These things can also be identified. So, from the fingerprint mass, because for many proteins the mass spectral digestion products by mass spectrometry are known, and that information is available as a database. So, that is for pure proteins, these are all available. Now, you take a mixture, you do the similar kind of thing, then obviously you can compare what kind of a fragments are more enriched, and then you go try to map with the known things in the literature, and from that you can say to reasonable extent, oh, this particular mixture has got this protein, then you can get their ratios as well. Then if you wanted sequencing, you can further take that particular protein and further fragment, further break it using a specific enzyme, and then you can sequence that too. And that is what is basically written in this. You can also find a lot of proteins will go through kind of a post-translational modification. Most often these are the glycomoieties, because these glycomoieties have got cell surface properties to attract something, not to attract something. So, therefore, that can also be identified from the mass spectrometry. A general methodology is you have taken cells or tissue and then make into thing and get a protein mixture, and you can separate them, and then you can digest, and then you can put this mixture, peptide mixture, through the chromatography, get this some separation, put into the electrospray method, or you can put into the MALDI, but generally initially you will put into the electrospray. Then you get the ionized peptides, then you measure. So, you can see that the ionized one, you can get the mass. You can further collide these ones, specific ones, and then get further fragmentation, and then you can get more ions and you can measure. So, you can take one of them protein, further ionize and get it. You can take a mixture and look at each protein. So, this box will tell you different kinds of proteins getting into that. There very little fragmentation. Therefore, you can compare this with the data available and then say what kind of a proteins are there in this mixture. Then a particular protein can be further broken into by ionization, and then you can look into. So, all of these are there. One specific example is shown here using a particular kind of the enzyme. So, protease enzyme, there are variety of protease enzymes are there. Please have a look at this particular thing. So, you have a chymotrypsin, trypsin, endoproteinase, variety of things. So, the different proteases will break at different peptide bonds. That is what I mentioned earlier. So, if you take one example here, this is a trypsin which is more frequently is used, and this will break at lysine at peptide bonds containing lysine or arginine, etc. It will break, and this is one polypeptide and treated with the trypsin, and this is the lysine, it's breaking here, this is arginine breaking here, lysine breaking here, another lysine breaking here, like that. It is here, it broke after 5 amino acids, it broke after 6, this is after several, after 2, etc., etc. So, you can, this is a very specific enzyme, so it will break at arginine and the lysine. So, you can get the fragments. Now, you look at the mass spectral details of this fragment and the intensities of this, and then compare with this data available in the literature. Already they are available for each protein. Then by comparison, if maximum amount of peaks of your spectrum matching with that in the literature means that particular protein, that is. So, for that you can get a figure of merit to what extent, 85% confidence, 95% confidence, 100% confidence, etc. can be obtained by comparing with this data and obtaining some kind of a figure of merit things. So, if you use the chymotrypsin instead of trypsin, this will break on the N terminal of the polypeptide, at the tryptophan, at the tyrosine, phenylalanine, leucine. These are the kind of things. So, one time you can do with the trypsin, one time you can do with the chymotrypsin, one time you can do another peptide if you want. So, at 3-4 different kinds of enzymes if you cleave, then you will get a data. Absolutely, you can perfectly make it. And you can do in each given protein the sequencing by doing the similar kind of thing. It's very well. So, you can find a protein in a mixture, you can find the, you can also do this particular amino acid sequencing as well. So, I have taken a few examples in general and specific. I said one for the trypsin, you can also do chymotrypsin, you can also do for endonucleases, many things known, they are known to break at particular peptide bond, that is the principle basically. So, everything else is an experiment and then mapping with the data that is available, identifying to what confidence level it is matching with that and these things. Okay, so I hope you understood the mass spectrometry in thing, particularly the electrospray ionization and the matrix assisted laser desorption ionization, which is popularly known as the MALDI. I have explained all of them. So, useful in large molecules, particularly in molecules where you don't want to break their association and where their clusters, etc., aggregates, etc., intact. Now, as usual, I used to give towards the end of each of the topic, I give some practice sessions, and this is another practice session for this based on the mass spectrometry. So, let us look at this. Take 1:1 EDTA complex of divalent cations of first row. The first row, you know, titanium, vanadium, chromium, manganese, iron, cobalt, nickel, copper, zinc, and divalent. And the EDTA, you take the 3 minus EDTA. So, that becomes 1 minus, because 1 metal and 2 plus, the EDTA 3 minus. So, therefore, total is 1 minus. So, that is Z is equal to 1 minus, and you can calculate, you can measure the spectrum for this, but here you are calculating, you are not measuring a thing. So, you calculate using that one, and you have to use the corresponding isotope distribution which you can get from the internet, and you do not copy any spectra which is given because they are available, all of these are available, but you calculate and give your spectrum, and for matching you can cross check with that. In this, you take the isotope distribution of the transition metal only, you do not need to look at the carbon, nitrogen, hydrogen isotope distribution, you can completely ignore, and then you draw corresponding calculated spectra, and if you are so enthusiastic, look for the corresponding spectrum which is known in the literature and try to match to what level it is matching. Okay, another practice session I have mentioned here. Collect the mass spectral data for a cell extract available, a lot are available from the literature, and see and practice. See means practice how individual proteins are identified from that. I have already mentioned the methodology just a while ago, and that is where the thing is. So, they always map with the with the mass spectral fragments with that maximum fragments matching will be that protein is presence is there. And then every intensity is you can tell you what percentage of that protein, what percentage of another protein, etc., etc. The query 3 or the practice 3, pick up one set of mass spectral data, anyone, for any protein to assign its sequence from the literature, a known case, and practice as how the data is utilized to assign the sequence. Because there it is the fragmentation at a known place. The known place means known enzyme. So that is where the digestion will come, and then you can see there the mass, and then from the mass of the bigger fragment with the lesser fragment you can identify the arrangement. Arrangement means that gives the sequence. So you can easily get already known. So take one of them, particularly you take something where the amino acid count is 100 or less, so that will be around 10 kilodalton or less. So smaller the better, easier you can make. So kindly do the practice so that whatever you understood can be really made to be utilized for your purpose of this. Okay, so this is useful for the master students who are doing their final year courses, PhD students who are doing their research both in organic, inorganic, polymer, materials, etc. Everywhere, all of them can be utilized. Even the researchers who are working in the industry, the R&D labs, etc., can be made use of this very much. Thank you very much.